Welcome back to Mad Medicine. In this lecture, we're going to be discussing eukaryotes. This is the part one of our lecture series on eukaryotes. If you haven't already done so, please subscribe to our channel because your support really means a lot to us. And if you want to support us even more, please turn off ad block because by you watching these stupid ads that come on these videos, you're able to support us and you're able to help us keep these lectures completely free for your educational purposes. If you want to help us even more, click on the ad. Okay, so that being said, let's just dive right in. Let's talk about eukaryotes. Eukaryotes are very important. These are types of cells you will be tested on and you need to know very well. So the main things, the general information about eukaryotes you need to know is that these are types of cells that contain membrane-bound organelles. Very, very important because this is a key distinguishing factor for eukaryotes. This is a key identifying aspect. These organelles. Okay, the reason why I say that is because eukaryotes do not have these organelles. Prokaryotes. I want to say this again. I want to say it clearly in case you got confused. Prokaryotes do not have membrane-bound organelles. Eukaryotes, which is the topic of today's lecture, do have membrane-bound organelles. I want to make that clarification because I just misspoke. Okay, so eukaryotes also contain a true nucleus with a linear DNA. These three components are very important because this would distinguish you from prokaryotes. Prokaryotes, let's write this right here, have no organelles. They have no nucleus, which makes sense because they don't have any organelles, and they have circular DNA. Okay, now there are 6 different types of organelles you need to know, especially in the context of eukaryotes, especially with their functions as well, and they are all listed right here. We're going to talk about each and every one of them in this lecture. All of these organelles sit in the cytoplasm. So with that being said, let's dive right in and let's talk about the cell. This is what the cell looks like. These are all the different types of organelles that are located within the eukaryotic cell. I probably should have written the eukaryotic cell. Okay, so let's talk more about these organelles. Membrane-bound organelles are essentially organelles that have a membrane around them. And the reason why this is important because it allows the organelles to be able to compartmentalize their function. They're able to function in and of themselves, so it is very segmented. This is an important factor for our cells because as you will find out, sometimes the functions of these organelles can actually be dangerous to other organelles if they were not closed off. That means you could be causing damage along the way as the functions were, happening. We'll talk a little bit more about that. I'll clarify that statement in a little bit, so just hang with me. And the other thing you need to know is that these organelles have membranes that are made up of a phospholipid bilayer. You have the hydrophobic head and the hydrophilic tail. What that means is that this is going to repel water, and this will attract it, which is a key distinguishing factor for any of the membranes we're going to be talking about as far as eukaryotic cells are concerned. And there is cytoplasm located both on the outside and the inside of the organelles. Okay? So, with that being said, this is what the phospholipid bilayer actually looks like. These are your hydrophobic heads right here, and these are your hydrophilic tails. In the actual membranes themselves, you have these channels where things can go through. They can be either diffusion channels or pumps. You have these proteins right here and carbohydrate structures that also are in the organelle. All of these structures have very, very important functions for the cell itself. Now, when it comes to the nucleus, this is the first organelle, that we're going to be discussing in the context of the eukaryotic cell. The organelle of the nucleus is very important because the nucleus is the control center of the cell. This is where everything is coming from. Think of this as the main brain of the cell. Okay, this is where everything occurs, especially in terms of telling the rest of the cell what to do. In the nucleus, you have genetic material, all right? The genetic material is DNA. So DNA sits in the nucleus. And the nucleus is surrounded by the nuclear membrane or the nuclear envelope. This is a double membrane that contains a nuclear environment that is separate and distinct from the cytoplasm. This is very important because this is going to allow for a safe environment. Why do you want a safe environment inside the nucleus? Well, because you have DNA in the nucleus and you do not want to damage that DNA. Now, you do know that DNA becomes RNA and then it becomes proteins, and then DNA and RNA actually has to leave the nucleus. How does that happen? Well, in the nucleus, you have pores in the nuclear membrane, and these pores allow for selective two-way exchange of the material between the cytoplasm and the nucleus. Very, very important because this allows for proper cellular function. You see, if the genetic material was not able to be moved from the nucleus, if it was not able to go from the inside to the outside, we would not really be able to do anything. We could not really control the cell. You need to be able to transmit essentially the commands that the nucleus wants. The mechanisms that need to be done by the cell have to be told, and the way it is told is essentially through these nuclear pores. They're going to allow for proper cell function because that will allow us to tell the rest of the cell, hey, this is what we want you to do. This is where it's saying it in the DNA. This is what the gene is being activated, etc., etc. This is a very important. This is a very, very important organelle. When the cell dies, the nucleus is going to disappear. So, cell death, one of the characteristic hallmark findings of cell death is no nucleus. Why do I say that? Because earlier I said that in the prokaryotes, they have no they have no organelles within the cell, and they have no nuclei. Well, if you get shown or if you get told that there is a cell without a nucleus in it, it could either be a prokaryote or it could be a dead cell, and you need to have more information, or you need to use the contextual clues to understand what they are talking about. So make sure you understand this basic concept of the nucleus. Okay, so in the nucleus, we said that you have this structure right here. This is the actual structure of the nucleus. Inside, you have the nucleolus, and you have all of this. You have the chromatin, you have the nucleoplasm. Remember, the nucleoplasm is not the same as the cytoplasm. Why is it not the same as the cytoplasm? Because you need to have a very distinct environment, a very specific environment in the nucleus. So it is not the same as the cytoplasm, whereas in other organelles, it's actually pretty much similar. The intra-organelle space is similar to the extra-organelle or the cytoplasmic space. You have these nuclear pores. These are the pores that we're talking about. So if this is the DNA and you need to remove an RNA sequence or you need to create a protein, it is going to leave from this pore. Similarly, if something has to go in, like a steroid molecule, like a hormone, it has to go in and activate a genome or whatever, or essentially activate a gene. It's going to go in through these nuclear pores. Okay, and this is a nuclear envelope. All right, so this is what the nucleus looks like, a general structure, and this is what one of the main things you also need to remember about the nucleus is that you have DNA in the nucleus, right? So the DNA itself is going to contain coding regions called genes. The genes are very important because those genes will synthesize, it will be essentially read, and they will produce from DNA, it will become RNA, and eventually it will become proteins. Now, the other thing you need to remember is that there is something very very important structure of RNA. You have 2 or 3 types of RNA actually. You have tRNA, you have mRNA, and you have ribosomal RNA. Okay? Ribosomal RNA is actually synthesized in the nucleolus. Remember that dark structure in the nucleus? So if this is your nucleus, this is the cell, this is the nucleus. You have a very dark structure right here. I'm going to write it right here. Nucleus, this is your nucleolus. Okay? This is where the ribosomal RNA is being synthesized. Now, the DNA itself is very important because the DNA is going to be bound in a linear fashion and is also supercoiled around histones. This is a defense mechanism. Why is this a defense mechanism? Because our DNA needs to be maintained. It needs to be kept safe. If there are any sort of damage to the DNA, it can lead to mutations and cause a lot of damage to the cells. So our DNA gets bound and it gets supercoiled around histones to make sure it is a very tight and difficult space for any damage to occur. Very important. And this supercoiling eventually leads to 23 pairs of chromosomes in each cell. Very, very high yield. Okay, so in each cell, a normal cell, you actually have 46 chromosomes, 23 pairs total. Okay, and of the 23, you have 22 which are just the normal chromosomes. Okay, and these are pairs, by the way. And you have one pair of sex chromosomes, either XX or XY. If you're a male, this is female. So female, male. Okay, pretty straightforward. This is what DNA looks like in the cell itself. As you can see, this is a chromosome, but if you dive deeper and deeper into this, you're going to see it's a very linear structure, right? And then you go deeper, you see these supercoils right here that are occurring. And if you go deeper into these, you'll see the double helix, but these double helices are actually bound, especially around these molecules. These are called your histones. When you get this structure right here, you will see a chromosome itself, and this is an image of what chromosomes look like. You have, remember, 23 pairs. This red is one pair, this green is another pair, and you have a total of 46 chromosomes all together. All right, so that is everything you really need to know about the nucleus, a good understanding of the nucleus. All right, so next we're going to talk about a very important organelle, similar to the nucleus, is one of the most commonly tested organelles, and that is the mitochondria. The mitochondria, as you may know, is the powerhouse of the cell, and it also contains two layers. Essentially, you have one outer membrane called the mitochondrial barrier, and then you have the inner membrane, or the cristae, and this is where the electron transport chain, etc., goes down. This is where everything is happening. By the way, this is a very high yield topic. You need to know where the electron transport chain occurs. It is in the inner membrane, or the cristae, of the mitochondria. And within those two layers, you also actually have two spaces. The first space is called the intermembranous space. This is the space between the inner and the outer membrane. And then you have the matrix, which is a space within the inner membrane or within the cristae. So I'm going to draw it, I'm going to show it to you, and then we're also going to see an actual picture of it. But let's say this is your mitochondrial layer, okay? This is your outer membrane that I'm drawing right now. And then you have this inner cristae. And the cristae are this weird shape. Okay. So this is your outer membrane. And this is your cristae. This is your intermembranous space. And this is your matrix. Okay, keep in mind that the cristae right here is where the electron transport chain goes down. All right, so we've talked about now essentially what the mitochondria looks like. One thing you need to understand about the mitochondria is that the mitochondria is semi-autonomous. They actually contain some of their own DNA and genes themselves, and these are called your mitochondrial DNA. Very important. And they also replicate independently because of independent of the nucleus because of binary fission. Now you might be saying this sounds very similar. This sounds like it is prokaryotic. Is it? Well, I'm going to give you another hint. Mitochondrial DNA is circular. So the theory is that at one point our cells were separate prokaryotes, and then the one prokaryote engulfed a mitochondria, and that is how eukaryotes started. Eukaryotes are thought to have evolved from anaerobic prokaryotes engulfing aerobic prokaryotes, essentially leading to the digestion, or the, I guess not digestion, but ingestion of mitochondria. And then from there on, over time and through evolution, we develop eukaryotes as we know today. Now, that's why you see this structure. That's why mitochondria have their own DNA that is circular. Okay, that's why you see that they replicate through binary fission, because that is what prokaryotes replicate through as well. So very, very similar. Mitochondria also contain another very important molecule, or very important thing that you need to know for your exams, and that is an enzyme. That enzyme is called cytochrome C, and it is located within, the intermembrane space. So if you don't remember, let's say this is, the mitochondria. This is the outer membrane, okay, and this is your cristae. This is the outer membrane. This is the intermembranous space. This is your cristae, or your inner membrane, and this is your matrix right here, okay? Here is where ETC, electron transport chain, goes down. And cytochrome C is located right here in the intermembranous space. Cytochrome C. All right. Why is this important? Because cytochrome C plays a very important role in a process called apoptosis, also known as programmed cell death. You see, if the mitochondria gets damaged, that means our body is not able to produce ATP. Okay, why? Because the mitochondria is the powerhouse of the cell. The main function of the mitochondria is to produce ATP. It is essentially the power plant of the cell. It allows our cell to function and allows our cell to be able to do the duties it needs to do by producing ATP and giving the cell energy that it needs. If you damage the power plant, you do not get ATP. When that ATP goes down, our cell is not able to do its jobs and it becomes defective. And when that happens, our cells know it's going to cost us more to keep that cell alive. So it's better to just kill that cell off and then move on and essentially cut your losses. One way that happens is that when you remove, or when you damage the mitochondria and you cause damage to this outer membrane, our cell knows that the mitochondria is now messed up. It's not working properly. The way it knows that is because cytochrome C leaves from the intramembranous space, or the intermembranous space, to the cytoplasm. And when it gets out into the cytoplasm, it will induce apoptosis by activating the apoptosis cascade, and the cell will die. That's why it's so important, and that's why this is a very high yield concept, high yield AF, in fact. So you need to understand this, and you need to remember this, commit this to your mind. You will not just be tested on this at one point in your education. You will be tested on this constantly. This is a basic concept of medicine and the human body you need to know. So now that we've talked about the mitochondria, let's look at a picture of this. This is what the mitochondria looks like. It's very tough to see on this electron microscope, but as you can see, this is your outer membrane right here. You can see this membrane. And then this is your cristae. Your cristae actually go like this, and you kind of can't see very well, but that's what it is. This is going to be your inner membrane right here, your outer membrane. Your intermembranous space is this region right here, and then your cristae is this, and this is your matrix itself, which is right here. Pretty straightforward. Okay, now let's talk about lysosomes. Lysosomes, think of these as the garbage men of the cell. The reason why is because their job is to remove, I'm just going to write stuff for the time being. Okay, they remove stuff, they destroy stuff, they get rid of it. And they contain this hydrolytic enzymes that actually break the stuff down itself. They get rid of the stuff by breaking it down. And these lysosomal membranes that are around the lysosome actually keep these enzymes from killing off the cell. Remember, we said that the actual cell organelles are compartmentalized, because if you are releasing the essentially organelle components, you might cause the cell to get damaged. Well, the reason why I said that is because the lysosome is the quintessential example of that happening. Inside the lysosome, you have these very, very important enzymes, but these enzymes are also very dangerous. So if they were just roaming around in the cytoplasm, the whole cell would die a lot faster. We don't want that, and the cell doesn't want that. So we have essentially put a barrier around them, and we call that barrier the lysosome. When we need to destroy something in the cell, the lysosome, we're going to call this the lysosome, and let's say this is the object. The lysosome will then come and actually engulf the object like this, okay, and it'll have the object inside of it, and then the enzymes will attack it, and that object will eventually break down and go away. That's how important the lysosome is. So what do you think would happen if you damage the lysosome right here? What do you think would happen with the enzymes? The enzymes would leave, and if they left and they got into cytoplasm, they would cause destruction, right? They would destroy the cytoplasm. And when they destroy the cytoplasm, what would happen? The cell would get damaged. And what happens when the cell gets damaged? Apoptosis, aka programmed cell death. And that's exactly important. That's exactly what I'm trying to get to. Lysosomes can be released to induce apoptosis as well, and they play a role in the mechanism of apoptosis. Cool. So we got through lysosomes. These are the garbage men. Next, we got peroxisomes. Peroxisomes are organelles that actually contain hydrogen peroxide. The name gives it away. Hydrogen peroxide is it's a very dangerous molecule. It causes damage, but it's important for you to have because you need them in order to do a lot of the functions. It's essentially, peroxisomes will also metabolize waste along with the lysosomes. So think of peroxisomes, in my opinion, as the garbage truck. Can't spell. Garbage truck. They assist lysosomes in removing this waste that we have, especially the metabolic waste. They play an important role in beta oxidation of very long-chain fatty acids, and they they essentially are created in the synthesis of phospholipids. All right, very important. High-yield concept. I think we covered peroxisomes pretty well. Let's move on to the endoplasmic reticulum, one of the most confusing of the organelles, but also one of the most important ones that you need to know. And we're almost done with this lecture, so just hang in there. So the endoplasmic reticulum is a series of membranes that are attached to the nuclear envelope. So essentially, remember, this is your nucleus right here. You have these little pores in the nucleus, right, in the nuclear envelope or the nuclear membrane. So let's draw it like this. Okay, you got these little pores. Cool. The endoplasmic reticulum actually comes off of the nuclear envelope. And this is what it looks like. It's a very distinct look that you're gonna see. Okay? It's like this. It's a very interesting look for the endoplasmic reticulum. There are two main components to the endoplasmic reticulum. The first one is a rough endoplasmic reticulum, which contains ribosomes. And this allows for the translation of proteins that are going to be secreted. So the proteins, essentially the DNA, will come out as mRNA or messenger RNA. And the ribosomes that are on this surface will actually produce or translate this messenger RNA into proteins. And it's called the rough endoplasmic reticulum because there are ribosomes and it makes it look rough. Okay? The second part is the opposite. It's a smooth endoplasmic reticulum which does not contain any ribosomes. There are no ribosomes in this. And this is important because the endoplasmic reticulum has two functions. Number one is going to be protein synthesis. Okay, this is through the rough endoplasmic reticulum. Number two is actually going to be a series of functions, which are lipid synthesis, detoxification, and transportation. This is all done by smooth endoplasmic reticulum. Okay, so I classify these as the other functions. One of the most important ones is detoxification. Where else in our body do we have detoxification occurring? If you recall from anatomy, it is your liver. Your liver plays an important role in detoxification. Well, as you can deduce then, the liver cells actually have a higher rate of the smooth endoplasmic reticulum than other cells because they need to play a role in detoxification. Just a fun fact for you. Okay, so smooth endoplasmic reticulum is going to help with lipid synthesis, with detoxification, and transporting the proteins from the rough endoplasmic reticulum to the Golgi apparatus. What is this thing? What is the Golgi apparatus? You might be wondering. Well, this is our last organelle that we're going to discuss, and we're going to talk about it right now. The Golgi apparatus is a very interesting organelle. This is what we like to call the post office of the cell. This is where all the proteins will go. Okay, the proteins will go there. Now, the endoplasmic reticulum will produce the proteins, right, with the rough endoplasmic reticulum. And then remember, we need to actually send these proteins out, and they need to be sent out by being packaged. And they get packaged in the Golgi apparatus. So the things from the endoplasmic reticulum are actually going to come to the Golgi apparatus in vesicles, and they're going to be unpackaged. They're not going to be set up. The Golgi apparatus isn't going to modify them by adding carbohydrates, sulfates, and phosphates. Why do you want to do that? Is because you want to get through the cell membrane. And you want to maximize the effect / the work of the protein. So you will be modifying those components. Then once the modifications are done, the Golgi apparatus will repackage them and send them to the proper location they need to go, whether it's intercellular, meaning within the cell, or extracellular, meaning outside of the cell. Whatever it is, the Golgi apparatus will send it that way. If a product is destined to be secreted, then the secretory vesicle will merge with the cell wall and actually release its components via a process called exocytosis. In exocytosis, think of it this way. Let's say this is your cell wall right here. This is the cytoplasm on the left side. And on the right side, you have the extracellular space. The Golgi apparatus will send whatever product we need in a vesicle. This vesicle will go to the cell wall right here, and then it will combine with the cell wall like this. Okay, I'm gonna color this red. It will combine with the cell wall, and it will essentially cause a hole in the cell wall, which will release the components it needs to release outside via exocytosis. Very, very straightforward. So now that we've covered all of essentially the high yield membrane bound organelles you need to know, we're going to stop here. Remember, make sure you understand the function and the aspects of a nucleus very well. Make sure you go through the mitochondria and know where everything is occurring in the different spaces, as well as the different components and where essentially processes are going on. And then make sure you know the rest of these organelles and what their functions are and why they're so important. Very, very well. Okay. With that being said, thank you so much for watching. I hope this was helpful. I hope this was educational. If it was, don't forget to subscribe to our channel because your support means a lot to us. It allows us to keep this content free. And if you want to see more educational content like this, go to our website, www.madmedicine.org, where you can find a bunch more free educational content because we know you're going in debt and we don't want to make you go deeper into debt. Thank you.